everyone. My name is Kate. Thank you for the introduction. Today I'll be telling you about this really cool collaborative project that I was part of, where we use deep learning models to try to predict changes to the ribosome that would stabilize it. And this work is in part funded by the NSF Center for Genetically Encoded Materials, also known as CGEM, and it was enabled by collaborations within the Innovative Genomics Institute here at Berkeley. So the ribosome is this amazing molecular machine. It's responsible for making all the proteins in the cell. And as an RNA biologist, I find this process really fascinating because most of the components involved in making proteins are RNAs. You have the mRNA, the messenger RNA that encodes the sequence of the protein to be made. You have tRNAs that carry the amino acids or the building blocks for proteins. And then you have the ribosome, which is mostly made up of RNA. And it catalyzes this chemical reaction shown here that links one amino acid to the next one in order to create a protein or a chain of amino acids. So at CGEM, we like to think about the ribosome as the proteins as a sequence-defined polymer. So amino acids are linked together to form this protein polymer, and the sequence is defined by the message in the mRNA. And in the cell, there are 20 different amino acids naturally occurring. And they all share the same chemical backbone, but they differ in the chemical properties of this R group here. And so by making different combinations of these 20 amino acids, life is able to create a huge diversity of proteins ranging from, say, myoglobin, which carries oxygen in your muscles, to GFP, or green fluorescent protein, which is found in jellyfish and glows green. And several decades ago, researchers began wondering if maybe we could add even more chemical diversity to proteins by incorporating unnatural amino acids that have unusual side chains that aren't found in nature. And dozens of these amino acids have been successfully added in specific places in certain proteins, allowing for features like site-specific chemical tags or labels. But in CGEM, we like to dream even bigger and imagine what if we could use the ribosome, not just to create different types of proteins, but completely new types of polymers with different underlying backbones. And so these polymers could yield things like therapeutics that are resistant to degradation, or new types of plastics or other materials with the sequence definition of proteins. So for example, one new monomer that could be really interesting to add is called a beta amino acid. So that's shown here. And a beta amino acid is similar to a normal alpha amino acid, but it has this extra carbon in the backbone. And so polymers created from beta amino acids could have protein-like functions that could be useful for therapeutics, but be resistant to the mechanisms that cells use to degrade proteins, extending the half-life of these therapeutics. But unfortunately, the normal wild-type ribosome is not good at making polymers from beta amino acids. And there's been previous work selecting for ribosome mutations that are better at using these beta amino acids. And that led to the creation of what's called the P7A7 ribosome, which has several mutations to the peptidyltransferase center of the ribosome. So that's where the polymerization reaction takes place. And it's slightly improved in using these beta amino acids. Using cryo-electron microscopy, some former lab members actually saw the structure of the P7A7 ribosome, and they found that it actually has a pretty big problem. This big chunk of the ribosome here, which includes the peptidyltransferase center, is actually disordered and misfolded. So it's not present in this cryo-EM density shown here because it's disordered. And this misfolded region comprises part of the peptidyltransferase center. So it seems like the mutations that might have improved beta amino acid incorporation also just cause this whole region to misfold. So this, as Eterna players might be familiar with, sometimes you make small changes to an RNA region and it unexpectedly causes the whole structure to unravel. So this is a bit of a problem. And to approach this challenge in ribosome engineering, we took inspiration from protein engineering. So suppose you have some protein and it is stable below some temperature, but unfolds if you heat it up. If you add mutations to this protein, like to engineer a new function, let's say, you might end up destabilizing this protein so it unfolds at lower temperatures. So that's kind of what we saw with the P7A7 ribosome. But what if you started off from a more thermostable starting point for engineering? Then, as you add mutations that add functions you want, this protein will still remain folded at reasonable functional temperatures. So this is kind of the idea that we wanted to apply to the ribosome. A workforce in both laboratories and industrial settings is E. coli. So we wanted to take the E. coli ribosome, develop a more thermostable version of it that could serve as a platform for further engineering. An approach that we've taken in the past is to look at these big databases of ribosomal RNA sequences from across the tree of life and look specifically at sequences that belong to microbes that live at extremely hot temperatures. So they're known as hyperthermophiles. So for example, these hot springs in Yellowstone. But unfortunately, you can't just like directly take the RNA sequence from these microbes and put it into E. coli. There's too many interdependent parts. It's just not going to work together. So what we did is we look at lots of ribosomal RNA sequences belonging to different hyperthermophiles and try to figure out specific individual changes we could make to the E. coli ribosome that will make it more stable. And we thought that this actually might be a suitable application for machine learning. We could feed a machine learning model sequences of ribosomal RNAs from thousands of different microbes that live at different temperatures, and then try to see if we can predict specific changes to the E. coli ribosome that will increase its thermostability and make it a better platform for engineering. To bring together this diverse team needed to approach this sort of challenge, there actually was a hackathon organized at the Innovative Genomics Institute at Berkeley. And participants included computer scientists, mathematicians, biochemists from multiple different labs and different departments. And we all came together for one day to brainstorm how to best tackle this problem. And at the end, a core group of us ended up continuing work on this effort for following months, accumulating in this bioRxiv preprint. And what I want to highlight is just like what a unique collaborative effort this was. So notice this star right here indicating authors that are major contributors to this work. And notice that we have 8 authors with this star. So this is kind of unusual for academic papers. So our first challenge that we tackled was how to get training data for these machine learning models. And we wanted a collection of RNA sequences that was both very diverse, so we have lots of training examples, but also minimally redundant. We didn't want the training data to be overwhelmed with a lot of the same sequences. And for this, we turned to the Genome Taxonomy Database, or the GTDB. So the GTDB is this collection of pretty much all of the available bacterial and archaeal genomes, comprising over 400,000 genomes, which is the version that we use. And many of these genomes are belonging to microbes that actually can't be grown in the lab. They're only known for like environmental sequences or microbiome sequencing. So they're very phylogenetically diverse. And the GTDB clusters these genomes into representative species and then organizes these species into a unified phylogeny. So we thought that we could use the GTDB as a framework to link both RNA sequences that are derived from the genomes to phenotypes such as the growth temperatures of these microbes. So these 400,000 genomes reduced to around 85,000 unique species clusters. And then for each of these genomes, we mine them to find representatives of 231 RNA families using covariance models from the ArcM database. And then we also used a machine learning tool called Tome to predict the optimal growth temperatures for each of these genomes. So we had both RNA alignments and growth temperatures linked in cross-reference to the same genomes. And together, this data comprised what we named Garnet. So it's a data set for training our machine learning models, and it stands for GTDB acquired RNA with environmental temperatures. So one of the first things we wanted to evaluate is if the collections of RNA sequences in Garnet actually represent a wider variety of sequences than the existing state-of-the-art databases. So here we're looking at all the RNA sequences in Garnet for a single type of RNA called the T-box leader, which is one of the 231 RNAs that we mined. And here I'm showing the number of sequences that we have for the T-box leader versus state-of-the-art comparison, which is the RFAM full alignment. And we're looking at the number of sequences at different identity thresholds, meaning that if we remove all the sequences that are more than, say, like 80% identical, how many do we have left? And here you can see that the Garnet alignments are deeper and more diverse, and that this depth maintains even if we go to like less and less similar sequences. And this pattern holds if we look at different types of RNAs. Here's just 6, for example. In the bottom are the ribosomal RNAs. And the only RNA family where an existing data set is much deeper than what we have is for 16S RNA, which is kind of special because it's been used extensively in barcoding environmental samples. So our next step was to associate each of these GTDB species with a growth temperature. So we had access to 2 previously existing data sets called Gosha and Tempura, which compiled existing information for several thousand microbes that could be grown in the lab. And then we also predicted the best growth temperature for each GTDB species using this machine learning method called TOME. And we did this because, as I mentioned, a lot of the GTDB species aren't cultivatable, so we don't have information in these databases. And these plots just show that the predicted growth temperatures from TOME correlate well for with the more established databases, Gosha and Tempura. So here is a phylogeny of all of the archaea in the genome taxonomy database. And these concentric wheels are plotting the growth temperatures in Gosha, Tempura, and then the predictions from TOME. And notice once more that the Gosha and Tempura predictions, it's like missing data for most of these organisms. You see this gray. So we have only like sparse and limited data available. Whereas for TOME, obviously we have a prediction for all of the genomes. And you see that organisms that were previously known to grow at really high temperatures, so they're kind of bright red, is also bright red in the TOME prediction. But the TOME predictions also include some clades of thermophiles that actually aren't included in these other databases and possibly previously unknown. So, you know, just to kind of sanity check that these predictions are correct, we took a look at the isolation sources for each genome. So this is something provided in the GTDB. And so on the GTDB, most species have genomes have an isolation source associated with it that says where this sample came from. And so if a predicted new thermophile is coming from an environment like a hot spring or hydrothermal vent, then we can be reasonably sure that that prediction seems correct. Versus if it came from an isolation source like sand or nasal swab, then maybe it's not actually a thermophile. And so we labeled all of the isolation sources as thermophilic or not thermophilic, and then looked at the correspondence of the TOME temperature predictions with these isolation sources. So you can see that the species that had high predicted temperatures tended to be enriched for occurring in thermophilic isolation sources. And this even holds true for species that are in what we call new thermophile families, or just like don't have any closely related relatives that are thermophiles in one of these established databases. So now that we had kind of all this data accrued, we set out to design machine learning architectures that could learn features of these thermophilic rRNA sequences. And one of the models that we designed was a language model. So you all might be familiar with ChatGPT, which is a type of large language model. Language models are also, of course, used in the foundation of protein structure prediction models like ESM2. And so we built one model that was a language model. We also built graph neural network-based model that I'm not going to describe the architecture of. But one more thing that I want to talk about is how we decided to encode the sequences. So sequences are typically tokenized before they're fed into a deep learning model. And we tried 3 different tokenization schemes. We tried fitting in the sequences directly as they were, tokenizing the nucleotides. We tried tokenizing the nucleotides in overlapping sets of 2, such that each nucleotide can be represented 4 different ways based on what's subsequent. And we also tried a triplet nucleotide encoding, where a nucleotide depends on both what's behind it and what's in front of it. And what we found was that the triplet nucleotide encoding worked significantly better for training our language model on RNA. And this kind of makes me wonder if, whether this kind of reflects what we know about the importance of base pairing in RNA structure. As I'm sure many of you appreciate, the stability of an RNA helix depends heavily on the 2 nucleotides on either side of it. So we wondered if the improved performance of the model with a triplet encoding might be reflecting biophysical correlations present in real RNA sequences. So this was a very interesting finding. Then to keep, how do we actually train our models? Now we have our architecture. Our plan was to initially train the model on all of the different types of RNA sequences from the GARNET database, from organisms that live at all different temperatures, so other types of RNA and ribosomal RNAs, so that the model could learn general properties of RNA sequences. And then we could fine-tune this model on ribosomal RNA sequences from 381 species that were predicted to be hyperthermophiles in the genome taxonomy database using this tone prediction. So then we had 2 different trained models, right? So we had this pre-trained model on all sequences and this fine-tuned model on thermophiles. And we generated 1,000 sequences of 23S rRNA from each of these models, and then compared these sequences looking for changes that are unique to the fine-tuned model. So it could indicate positions that are important for stabilizing the ribosome at high temperatures. So how do we first do basic quality control to see if these models are producing sensible outputs? So for just like as an example, suppose we're using some deep learning model like DALL-E to generate an image from the prompt. So the prompt is photograph of 2 hands shaking, and then we can just visually examine the output image and see if there's potentially something wrong with the model, if we can really trust the kind of outputs it makes. So we can do things like look at the number of hands and look at the number of fingers on each hand. But what about our model, this RNA language model? We prompted with the first 100 nucleotides of the E. coli 23S rRNA sequences. It kind of completes the rest of the sequence. And it looks like this. And so just by looking at this, can you really tell if this is like a sensible output or if this is like a hand with 3 fingers? So we did a RNA equivalent of kind of figuring out what's in the sequence. So the first thing we did is we took the 1,000 generated sequences from each model and we assessed this CM search score. So this is essentially a score that says how well do these sequences fit into a pre-existing covariance model of what 23S rRNAs should look like. And you can see that the generated sequences, this blue distribution, they're kind of in line with the range of CM search scores that naturally occurring sequences produce from the GARNET database. So these kind of look about as much like 23S rRNAs as naturally occurring sequences do. So that's good. Another thing we looked at as a quality control measure is whether these RNA helices in these generated sequences had how many non-Watson crick or non-GU base pairs it had. So essentially, how many non-canonical base pairs are there? And we thought that if this model is just spitting out nonsense sequences, then these sequences might have a lot of mutations in them that disrupt the expected secondary structure of the 23S rRNA. And these plots show that these generated sequences, these blue distributions again, have a similar fraction of non-canonical base pairs in their secondary structure as naturally occurring sequences. So this is kind of, the equivalent of counting the number of fingers on these RNA sequences. Our next goal was to find specific positions in the 23S rRNA that the models might have learned as to be important for thermostability. So our idea was to look at each individual position in the 23S rRNA and ask, how does the distribution of nucleotides in the 1,000 generated sequences differ between the trained model and the fine-tuned model? So we thought that if a position is not important for thermostability, then these distributions should be similar. But if it is, then the fine-tuned model, which was trained on hypothermophilic sequences, would be different. And we quantify the difference between these distributions using a measure called Jensen-Shannon divergence. Here I'm plotting the top 200 most divergent positions on this kind of road pill splatter visualization of the 23S rRNA secondary structure. And the potentially thermostabilizing mutations are marked in red. And I just want you to notice that these are kind of distributed all throughout the sequence. Of course, they're kind of missing from the beginning of the sequence because we use that region as the seed. And also that a lot of these top mutations occur in base pairs. So with one occurs, the other occurs as well. So telling us the model is learning, at least on some level, something about base pairing. So we, as part of CGEM, we're interested in stabilizing the peptidyltransferase center of the ribosome. Remember that region that was unfolded in the P7A7 ribosome? So we're interested in mutations from this model that could potentially stabilize this region and help with engineering. And the PTC is known to fold last during the assembly of the large subunit of the ribosome. It's in what's called domain 5, and you can see this red domain 5 kind of folds as one of the last steps. So we were kind of particularly interested in mutations in these later folding domains. And one thing that kind of stuck out to us is these mutations identified in Helix 89, which is pretty close to the PTC. And as I mentioned, we actually had like several different models that we trained, some with slightly different architectures. And it was nice to see that some of these mutations were actually independently proposed from multiple different models. And so these mutations, one concern that we had, because like up until this point, we're kind of assessing them individually. And we were worried that whether these sequences would actually work in the context of the E. coli 23S sequence, or whether these mutations depend on another mutation co-occurring. So to test this dependence, we introduced candidate mutations and kind of like made these synthetic sequences of the E. coli 23S with individual mutations introduced. And then we assessed the likelihood that the fine-tuned or the pre-trained model would have produced the sequence. So we're kind of asking how good of a fit is the sequence to this model. And then we compared the likelihood of these sequences being generated by the fine-tuned model relative to the pre-trained model relative to wild type. So essentially, the takeaway is that if this value is positive, it means that this sequence is better supported by the fine-tuned model, which is trained on hypothermophilic sequences relative to wild type. And we assessed these 4 positions that were mutations that were proposed in Helix 89. And you'll notice that these 2 loop mutations, 2474 and 2477, they have positive values. Their models support them as potentially thermostabilizing in the context of the E. coli sequence. But these 2 positions at the base of this helix are individually not stabilizing. So this kind of makes sense, right? We're introducing them individually. So this breaks this base pair. So it's not surprising that these mutations on their own aren't good. And we would expect that if we introduce them together, it would make them much better. But unfortunately, I couldn't find the data point for this presentation. So the next thing we wanted to test is whether these candidate mutations actually work experimentally. And we have this assay running in the lab to test the thermal stability of ribosomes. So the way it works is first we take the ribosomes and we pre-incubate them at either 37 degrees normal temperature for E. coli or at 65 degrees Celsius, which is really hot. And we see if these ribosomes are still functional in an in vitro translation assay. They can still translate this hybrid peptide. And if they are thermostable, then they can withstand this 65-degree pre-incubation and still be functional afterwards. So we tested these loop mutations. We tested 2474, also in combination with a nearby nucleotide, and we tested 2477. And you can see that all three ribosomes, the wild type and the two mutants, have similar activity after 37-degree pre-incubation, but after being exposed to heat treatment, only the 2477 mutant retained a lot of activity. And meanwhile, the 2474 mutant was not stable. We don't fully understand why. It could be a variety of reasons, but at least this one looked really good. We're similarly interested in this other helix. It's called the helix 68, and it's located in domain 4, which is another late folding domain. And many of the models suggested mutations to this intriguing set of non-canonical base pairs. And so these mutations would kind of turn them back into normal GC pairs. And we were really curious to see if these would actually be stabilizing in the E. coli ribosome. So we did the same thing where we assessed the model probabilities for these mutations for these three base pairs, both individually and the doubles. And you can see that all of them appear stabilizing both independently and in combination. And we also assessed all three of these base pair changes all together. And doing the heat treatment assay showed that these ribosomes are actually more than twice as active than wild type after heat treatment. So these mutations are also thermostabilizing. So our models are helping us identify sets of mutations that actually stabilize the ribosome in the lab, which I think is really, really cool. And that's kind of where this project leaves off. Future directions include testing additional 23S RNA mutants that our models suggest. We also want to see if introducing this helix 68 and helix 89 mutants could stabilize and restore the P7A7 ribosome. And even more further in the future, we hope to use Garnet to train additional types of models, linking other types of RNA sequences, RNA sequence features to functional phenotypes. And with that, I'd like to thank all of my co-authors, this multidisciplinary team that made this work possible. In particular, thanks to Amos and Jamie for sharing a lot of the slides with me. I'd also like to thank other members of the Kate Lab for their support, the Center for Genetically Encoded Materials for funding and support, and the Innovative Genomics Institute for enabling this really cool collaborative project to take place. And so with that, I'm happy to take any questions.